Everyone, you just entered the second webinar of NETRI, talking about traumatic nerve injury and platform and innervated skin. I think we have to wait a few seconds to get the go live, and then we will move to the presentation. So stay with us. Hi, welcome everyone for the next webinar that triggers the second session of this program from NETRI, and we will talk today about traumatic nerve injury platform and innervated skin. So I hope everyone is listening and is ready to hear about the solutions and the application that we can present to you. So first of all, I will briefly introduce the two speakers of today, Hélène Gauthier, Dr. Hélène Gauthier, PhD in neuroscience, and Alexandre Guichard, also PhD in pharmacology. They will present themselves a little more in details when they will take the speak. So today, as a program, we will talk about, in first, the peripheral nervous system application that Dr. Hélène Gauthier will introduce to you. And then we will move to the neurocosmetic application from Alexander's talk. And then we will have a question and answer session. So just as a reminder, the proposition of the NETRI technology is based on a very easy device to use, high throughput, and also without pumps. So that means that the culture of the cells will be done directly using our device without pumps, and it's basically a standalone device as we propose to the market that will go into the incubator. The other things that we will briefly remember or recall for the audience and also for the new participants that have never heard about NETRI yet is the compartmentalization. So we will talk to that in the next slide. Finally, the way we have designed the devices that we will talk about are based on a full compatibility as it is a 96 well plate. So lab equipment will be suitable for characterizing through the devices. The other thing that we will mention during our talk is the ability of NETRI technology to biodigitalize the body, and so to be able to innovate the different organs of the body. So we'll come to that, and that will be a response to the market needs in terms of readouts that is needed, including the electrical activities, electrophysiology, or also called MEA, as well as the capabilities to have dynamic and non-destructive responses with our devices. So this is an important slide. I would just like to step down for 1 or 2 minutes. Is to understand why NETRI technology is so unique. And if you understand this slide, I think you will definitely be able to understand the power of the technology and the application you can get from it. So as you know, it is commonly used to use the well, like transwell, to have studies on different mixed cell types and to try to modelize the behavior of those cells during the growing of the cells and testing the cells through this well. This is, of course, a very non-expensive characterization way and technique. However, it has limitations, and we know the limitations. So we basically, the market is trying to propose to the users some devices or platform that are capable to mimitate the human body. And that's basically what we are doing in our technology. So as we talk about the way we produce our devices, first we produce our devices by 4 chips, which is the unit of production here, and then we will come to what is exactly a chip, a netry chip. So basically, a chip is composed of 6 wells and 3 channels. So the channel 1 here, that if you follow my mouse on the left, then the second channel that is basically vertically separating also channel 1 from channel 3. So as you can see, there are 2 wells per channel, which means there is an inlet and an outlet of each channel. So when you introduce cells into the channels, then they will migrate into the region of interest here, and then you will be able to change and replace the media by a proportion, different proportion, between 50% to 100%, depending on the cells. But however, you have the ability to pipette in and pipette out. This gives you the ability also to collect supernatant and make some, for example, chemical analysis, generally speaking. So the concept of our technology is that the middle channel is basically acting as a physical wall through the design of our chip. That means that it is capable to avoid the migration and diffusion of liquid from channel 1 to channel 2 and from channel 2 to channel 3. So the result of that is the ability to do co-culture or even 3 culture into one single chip. So the culture will grow separately. So that's the first thing. Additionally to that, we have introduced microchannels, which are here basically represented, which are extremely small. It's like microchannels of about 3 by 6 micron size and lengths of about 150 to 450 micron, depending on the architecture. And those microchannels are here to allow the projection of the neurons, so the axons, to grow into those microchannels. They will be guided into this channel and will be able to go from the channel 3 then to the channel 2, and then there will be again microchannels that will guide the projection of the axons into the channel 1. The result of that is the capability in our devices to do innervation of organs. If we take here, for example, keratinocyte cells on the left, which are represented in red. We can also do neuron to neuron if we want to address the NDD models and studies. So this is probably the most important picture here of the technology, that if you understand this, then you understand that you're capable to study the growth of sensory neurons into other compartments, which could be other neurons or other organ cells. There are some capabilities also behind the technology. Is the ability to do electrical measurement of the activities of the neurons and the network of the neurons, and that's something that will be actually illustrated through the presentation of Dr. Hélène Gauthier. Finally, I would like to say that the frame of the device that we propose is basically, as I said, a 96-well format, which helps the compatibility of the lab equipment. And additionally to that, the PDMS, which is the material we use, which is a type of silicon for manufacturing our devices in an industrial process, helps to be transparent. So that means that we will be able to characterize from an imaging standpoint. as a summary of our last webinar on CIPN on chip, we've discussed basically results from the ability to grow neuronal cells into our devices, and using different compounds and testing, for example, oxaliplatin or paclitaxel, and being able to address through axotomy the capability to grow the neurons.
This one.
Okay, so this is something that you would be able to see on the replay on your YouTube channel, and basically would be able to understand how to use the strength of the compartmentalization of the NET3 devices in CIPN applications. I would like now to introduce you the next speaker, Hélène, which will talk about the peripheral nervous system application.
Hello, everyone. So I'm Hélène Gauthier. I'm a neuroscientist specialized in developing functional assays based either on electrophysiology or imaging. So I have experience in pharmacology, toxicity assays, and I joined NET3 nearly 2 years ago now. So I will briefly remind you about the anatomy, this real compartmentalized anatomy of the peripheral nervous system. So on the left side of my screen, you will have the representation of the skin, where you will find free nerve endings, nociceptors, different type of sensory neurons and endings, and that will the axon will travel, and the cell body will be located in dorsal root ganglia, and they will have also axon projected to dorsal horn neurons. On the other side, motor neurons are located on the ventral horn and are projecting towards the periphery, where they will contract muscle at a really specific synapse called the neuromuscular junction. So this was just to introduce regarding this compartmentalized anatomy, how it fits with the compartmentalization of NET3 chips. And today I will explain how we can mimic or reproduce some type of traumatic injury in the chips. So there's many situations in which a traumatic injury in the spinal cord or nerve can induce neuropathic pain, and it will also induce debilitating motor and sensory symptoms. And it can be following different accidents at work, or during activity like some sport. It could be during surgery, or it could also be compression by a tumor or a carpal canal syndrome, as an example. So some injury, there's different grade of injuries, and there's also a different type of treatment. And the ones that will really have an important impact on the quality of life are either regenerative medicine and attempt to reconstruct or regenerate nerves. They can be either surgical or non-surgical, and especially in the non-surgical approaches that NET3 chip can be really helpful to develop treatments that can be based on growth factors, on gene therapy, and to test this in vitro on human cells. And what will really help patients is efficient pain relief. So in order to mimic human nerve trauma and also to be translational and be a bit a mirror of current in vivo models such as nerve crush injury or ligation, we use an NET3 chip. We've been able to show that we can do repeatable and standardized injury to target only neurons and not the cell body. That something can happen if you are just in a well environment, and also that we can recreate microenvironment and potentially regeneration environment. So regarding our the culture, so here you have example of different architecture of chips. And what I will present you can do it using either the DuaLink Shift or the DuaLink Delta Ultra. The main advantages is really to have the soma in one compartment and to have long projection in the other compartment. in order to be able to screen for drug candidates, to look at growth kinetic monitoring, and also to combine either electrophysiology, imaging, and biochemical analysis with the same experiment. A little reminder of how our device look like. You can have two versions. Either the light version where you have 8 chip and data point per plate, ideal when you're starting experiment and don't want too many in one go, or the 16 chip in the full version. Now let's talk about the cell culture and how we adapted culture of hiPSC-derived motor neurons in our chips. So this is what I wish today we've been using motor neuron from Fujifilm. So this is the data from the supplier. What we did is to test the initial supplier protocol and adapt it to fit in our chips. So we adapted and changing coating compared to 96 watt plate in order to have a repartition of cells that will promote the neurital growth and have a good repartition of the growth in the chips. We ensure that the cell was still expressing markers of motor neurons, such as islet 1 or cholinacetyltransferase, as the supplier showed, and also shows expression after 21 days in vitro of maturation marker and reduction of pluripotency markers such as adenosine. So we do have, for the also NeuroFluidics line, a fully validated culture protocol. And we tested also the protocol on the DuaLink MEA, and we have our first proof of concept data and showing that cells are viable and can have bursting activity when they're seated. Here they're just seated in one channel and not projecting in the other one. Here's the data for the sensory neurons. So here the supplier is axonal. So you have again the data from the supplier showing presence of certain channels of relevant marker for the cell type or TRPV1. And what we did is to adapt the culture protocol and also validate for some of those markers. So here you will have an example of peripherin. TRPV1, and this is an example showing at 3 weeks of culture how the neurons can really project really far in the microchannels using a beta-3 tubulin testing. We adapted the protocol on the NeuroFluidics line and also on the MEA, and we have fully validated protocol on both substrates. And here is an example of staining expression of substance P in the devices, as well as the morphology of sensory neuron culture in DuaLink devices. And regarding the electrophysiology, we are able to QC each cell before treatment. Today, the topic is regarding axotomy. So what we did, we tested and we created a repeatable and standardized axotomy protocol by using a detergent application and a short one. So here you have the schematic of the DuaLink Delta Ultra that has been used in the studies of the time presenting. So you will have either the motor or the sensory motor, the soma in the channel 1 compartment, the axon projecting towards channel 2 and channel 3, and the application of the Triton in the channel 2. So this is, here you have images of the axotomy when it was performed at 7 days in vitro. So this is when the axons are in channel 2 and they start to go into the channel 3 at the top of the, On A to D are motor neurons. So here on bright field images, before and after the axotomy. I'm not sure you can really see very well, and I invite you to look at our application notes that will summarize this data. We tested the visualization with different live staining. So both were able to show that 100% of the neurites are absent 2 hours after detergent exposure. And to be able to look at the regrowth, we choose here's live staining that was giving us the best axonal detection, which is a calcine presented in those 2 panels. Here, example of the sensory neurons regrowth. So 24 hours after axotomy, you do have in the channel 2 that was completely devoid of processes 2 hours after the axotomy. You can see the regrowth. And if we look at 7 days after axotomy here, and you have a magnification in C, you can see that 50% of the chip have axons that are rich channel 3, and you do have a really good regrowth. And you can really follow up the axons and how they're regrowing. Now I will show you how example of this platform and how you can use it to test different compounds. So similar, it's exactly the same schematic of the DuaLink Data Ultra, using some motor neurons and applying 2 different mechanistic controls on the neurite extension. So nocodazole is interfering with microtubule and is used as a neurodegenerative molecule. Staurosporine is a protein kinase inhibitor and has been shown to be neurotrophic. So they were added in the axotomy channel 2, and we observed the regrowth 3 days after with the calcium live dye. What we noticed that here you have the images at the highest concentration tested of nocodazole where no signal could be detected. At 1 micromolar, there is really an inhibition of the neurite outgrowth compared to the vehicle application where you can see the lines of neurites inside the MAC channels. And the staurosporine at 50 nanomolar on the motor neurons enhance the neurite outgrowth. And here is the quantification that was done. As I mentioned, at 10 micromolar, we also had effect on the viability of the motor neurons, and it wasn't whereas the 1 micromolar only affected neurite outgrowth parameters such as the number of axon or the maximum length, and also this total sum of lengths. We could also, so with the staurosporine, we can see that the number of axon and the sum of lengths are increased, showing the neurotrophic effect of the staurosporine in this model. And I will take just a little sip. Before showing you regarding the sensory nerve, so same protocol and same application of compounds in the channel 2. Here we tested, so this time nocodazole at 1 micromolar was enough to affect the viability and completely prevent any regrowth, and in fact there was degeneration. So we tested lower concentration, 0.1 micromolar, that wasn't affecting viability but affecting the neurite outgrowth. Here you have the picture for the vehicle and for the staurosporine, and it was exactly the same concentration tested. Difference is that we observed only 2 days later because the sensory neurons have a faster kinetics of regrowth. And here's a quantification. So as I mentioned, we couldn't measure any parameters at 1 micromolar, and so we tested the nocodazole at 0.1 micromolar. We couldn't detect any significant effect of the staurosporine as a tested concentration, which in fact we show here how this platform can detect differences of efficacy depending on the cell model used. And I will conclude by how you can combine the electrophysiology with axonal outgrowth, and this will be by using the DuaLink shift MEA, because you have a long microchannel on one side of 50 micromolar and the electrodes that are positioned at different locations underneath the microchannel, so you can follow the electrical impact of axonal regrowth. And follow the electrical impact of neurotrophic compounds that you might want to design therapeutics that enhance outgrowth. But if it generates abnormal electrical activity, you could see it using those devices. And finally, you can ensure that you have a functional recovery after regrowth.
Thank you very much, Hélène, for the talk. Take some water, that's fine. We appreciate that. Please remember that there will be a Q&A session at the end. So we have already some questions coming as we speak from my marketing team here. So we will address those questions, obviously, in a few minutes. So let me give the talk now to Alexandre, that will talk about the neurocosmetics application, and I will let you introduce yourself and go ahead.
Thank you. Good evening, everyone. I'm Alexandre Guichard. I'm a PharmD, PhD, and I did research on cutaneous pharmacology, either clinical and in vitro research. And here at Day3, I'm in charge of developing the new application, a new product in regards with cosmetics and dermatological field. As you may know, the skin is innervated. And the skin is a protective organ, the biggest organ we have on our body. And this organ is able to beside its role to protect ourselves and our other organs, it's also able to decode a wide range of tactile, thermal, and noxious stimuli. And this is possible because of the interaction between keratinocytes and neurons. Both cells are connected and involved in many normal or pathological skin conditions, such as pruritus, sensitive skin, psoriasis, atopic dermatitis, eczema, herpes infection, or also wound healing and many other conditions. Nowadays, preclinical research are limited by in vitro models, which are not always relevant in our field in dermatology and especially in cosmetic, either because they use rodent neuron cells, which is not allowed in cosmetic, or because the co-culture, as explained before, are based on mixed cells, either skin and neurons, which is not a real models of in vitro model of the skin. It doesn't mimic the anatomical and physiological structure, because as explained just before by Hélène, physiologically the cell body of the neuron is in one compartment, in the ganglia, in the medulla, and the axon ending are located in the skin, into the skin. So two physiological compartments which we are trying to reproduce in our in vitro devices. Yeah, this is the slide you've seen just before. So I won't spend more time on this, but here the important thing is to understand the real interest and advantage of the compartmentalization. We can draw a list of these advantages, but I will just focus on three. First, by using two different compartments, you can use the specific medium of each cells. For example, we know that the keratinocytes, they like specific medium, specific ions that the neuron doesn't like, and the reverse also. That's why by using our chips, you can use the correct and the suitable medium for each cells. The second advantage of compartmentalization is the supernatant. If you sample the supernatant to analyze the secretome of your cells, you are sure that the cytokine or whatever you want to dose comes from this cell type and not the other. So you again have more relevant results of your dosage. And the third advantage of the compartmentalization is the treatment. When you put a treatment on the skin compartment, you have in this compartment the skin and the axonal endings, which is a real life. In real life, when you put a cosmetic product, you will put it on topically on your skin and not directly inside of your spinal cord, which is the other compartment with the cell body. So these are the three main advantages of the compartmentalization compared to, for example, a traditional well. I will just give you more information about what we call MEA, electrodes, electrophysiology. It's as introduced Hélène, it's electrodes directly inside of our chips. Our cells, the cells, when they are seated in our chips, they grow directly on the electrodes, allowing the assessment of their electrical activity. With our chips, for example, here you have three compartments, so you can it allows the co-culture of two different cell types, for example, and you have the channel here in green to increase the fluidic isolation by using a kind of wall of medium. These I will give you more information, but just after about the advantage of the electrophysiology and of our system. But just keep in mind that here today we are presenting you two different applications, but many applications are possible in neurocosmetic, in PNS disorders, but also in immunology. We can also imagine also the microbiota, the gut, and other application, oncology, and also as said Thierry, neurological disorders. To analyze the electrophysiology, we need specific devices, which is here devices manufactured by Axion, our partner. Our plates are compatible with this system, to assess electrophysiology, but with other systems. Before I give you more information about the electrophysiology, just you should understand that with one chip you can combine many readouts. Since our plates are compatible with the ELISA reader, with other traditional readouts such as live dead assay, immunofluorescence. As said Thierry, our material is transparent, so you can use confocal microscopy or the microscope. Liquid chromatography, you can lyse the cells to and collect the supernatant. Live staining, calcium imaging, and mass spectroscopy. And so the electrophysiology. This system is very interesting because it's not cytotoxic. So you can use it in addition to the other, in addition with what you used to do. And the other advantage of the non-cytotoxicity is you can assess, you can measure you can repeat the assessment on the same chip for different time point. You can also have a continuous recording for several minutes till 1 hour even, and this is very interesting, especially when you want to assess the effect of sequential treatment, for example. So it means that one chip, one condition contains its own control, because before you put your drug, you put also the vehicle. So you have your chips has inside of it the vehicle, so which decrease the variability, the inter-chip, inter-condition variability. With this device, you can also you can have a live monitoring of compound, especially compound with very quick effect, very fast effect. So you can, you can have a continuous measurement. You can every second follow the effect and the response of the neuron and your cells to this treatment. And also the advantage of this machine is you have electrodes, so we use them as a receptor of electrical activity of the neurons, but they can also be used as a stimulator. You can use the electricity of the machine to induce the excitability of the neuron. This is something the machine is able to do it. It's still under development at NETRI, so we don't use this module yet, but it's something possible. So which is very interesting, because you can have a physical stimulation. And we can also we have a digital team at NETRI who are in charge to develop specific software, and especially to facilitate the assessment and the analysis of the data. This is our two main formats: the H version with 8 chips with the electrodes, and the Pro version with 16 chips. Now the use case, a proof of concept of what we are doing, what we are still developing here on innervated skin 2D. So you've seen this figure before previously. So in channel 1 on the left side we see the neuron, and on channel 3 we see the keratinocytes, and the micro channel between the 1 and the 3 allow the axons of the neuron coming from the 1 and go to the channel 3. So we are developing the model, and we already get some first results. So some of them are confidential, so I cannot give you more information. But here you can see, for example, an example of a co-culture in immunofluorescence. You have basically in yellow the neuron and the axons, and in blue the keratinocytes. And you can see here the axons of the neuron projecting to the keratinocytes compartment. Here it's another example of what we did in another experiment. We recorded the electrical activity of the neuron after addition of a treatment which is supposed to have an hyper-excitability effect. So here, as I said just before, the chip it's its own control. Is its own control because, as you can see here, is the basal electrical activity of the neuron, and then you put the vehicle, so you have a small spike here, because by adding a vehicle, by adding something, you have an effect due to the flow of the medium. You have a mechanical effect on the mechanoreceptor of the neuron. So you have an effect. And that's why we do it first to take this effect into account when we consider the effect of the drug or the compound. Here we put the compound X, and you can see an hyperexcitability of the neuron, and then they come back to their basal state. And as you can see, this is very short. So this is that's why it's interesting to have this kind of reader with this detailed assessment. So we then at the end of the experiment, we always use a positive control. Here it's KCL, which depolarized in a non-specific manner all neurons, and this is our positive control. If this test at the end of the experiment doesn't produce any spike, it means that the chip is not this chip is not active, and so we don't take this chip. We don't use it for our analysis. So it's also an internal positive control. And then we can assess the percentage. We can calculate the percentage of change of mean firing rate, which is one of the several parameters we can have with this machine. This electrophysiology can be used either for acute or what we call chronic activity modulation. Chronic because it's not chronic, but in short time. In our experiment, we use several hours after the first application of the product. But we can also imagine treat the chip and then put it back in the incubator and read another time later on. It could be possible thanks to the non-cytotoxicity effect, non-cytotoxicity capability of this readout. And then so the applications of this model are broad in our field, either to assess the pathophysiology, so the mechanism of action of drugs, the pathophysiology of diseases, the pharmacology. It can also be used. Our model can be used as to screen efficacy or toxicity of your cosmetic or of your drug. And the application are also every application involving skin and neurons, meaning sensitive skin pruritus, inflammatory dermatosis, or on the reverse, well-being, antioxidant effect, for example, the wound healing. And today I presented you keratinocytes in monoculture, but we can also consider all the epithelium, cornea cells, buccal oral cells, vaginal cells. Every epithelium is actually close to keratinocytes. And why not you consider also personalized chips? So if you use, we can use cells from patient or from subject and primary cells, and maybe put in our chips to have a personalized chip for specific questions, for example.
Thank you very much, Alexandre, for the presentation. We will now start the Q&A sessions. Please stay with us. We have some surprise at the end of the Q&A session, so stay with us for the next 15 minutes. So it would be interesting for you, if you have some interest in network technology, to hear what we can propose at the very end. So that's the teaser of the webinar. There have been quite some numerous questions, so we will I will address them basically not in an order, just taking them, and I will ask Hélène and Alexandre, but I also have Thomas, which are joining us, as well as Jessica, which are technical authorities that will also possibly will support the questions. So the first question comes and talking about tree cultures. So, and the question is, can we have a third cell population in the middle chamber in DuaLink? I think we're talking about having tree culture actually in our chips. So maybe I can answer this question. I would say not too much scientific here, although I'm a scientific also. But yes, we can definitely do a tree culture in one chip. So the middle channel will be at the same size and volume that more or less the two other channel 1 and channel 3. So that's absolutely possible. And we will be able to give you more details later on. You want to add something, Hélène?
I will add so you talk about the similar size channels that for the DuaLink that has been adapted to have the tree culture, and there's larger channel just for this.
Yeah, absolutely. So another question that's coming now from the material that we are using, the PDMS. So it's a silicon type. The question is, since you are using PDMS to make the chips, did you check DMSO absorption into the chip? Is there someone that wants to answer this question from the scientific audience? Maybe Hélène, you are.
I will answer it. That we haven't tested DMSO directly. We did develop a protocol to be able to check absorption. But
So if I can add something there. We know that we have to be cautious with PDMS absorption, and that is something we are taking into account based on the physical property of the molecule, not necessarily DMSO, but the molecule you're interested into. We can have a look in the literature to see whether it is likely to be absorbed or not in the PDMS.
Okay, thanks, Thomas, for that. There is another question talking about the, basically the question is, can you tell us which protocol was used for the differentiation in sensory neurons? Hélène?
Okay,
Still?
I guess so we use commercial cells. So the axle in this case is taking care of most of the differentiation from the hiPSCs to derive the cell, and we are just buying them as progenitors as a final stage. It's based on chambers protocols, if that's the question. But we don't do the full differentiation in the house.
Okay, good. Now I will combine two questions that are talking about measurement of the electrical activity of the neurons, the electrophysiology. The first one is how many recording electrodes in the 3 compartments of a unit? I guess it's how many electrodes are composing one chip. I think that's the question. And within this, the same person, can some of these electrodes be used as stimulating electrode? I think some answers have been made already. So maybe I will let Alexander respond to that.
The number of electrodes is 46, 48. It depends. 48.
It depends on the architecture.
Yeah, it depends on the architecture. And also we, depending also of the application, we don't assess all the electrodes. We, for example, in some case, we use especially the electrodes below the microchannels because we know that this is a kind of concentration of the information, and especially the information is higher. So maybe you can
develop on this.
Yeah.
The physical structures of microchannels, making it a very small environment, is acting as electrical amplifiers. So we'll actually, thanks to our devices, record a lot more signal that what can be compared to standard MEA plates. Also, the microchannels also guiding the axons through them, so there is a very high chance of having functional axons on top of the electrodes. So those two, even combined, make it ideal to look specifically at those electrodes, and they will also represent the information, the signal going from one compartment to another. So this is very relevant information to look at.
In specific context. And for the stimulation?
So this technology is based on Axion technology. You can turn any of the recording electrode into stimulating electrodes. And you can turn 1, 2, or 3 depending on where you want to stimulate. I would not recommend to turn too many electrodes in stimulation, but yeah, it can be done.
We can imagine to turn the electrodes on the soma.
Or maybe 1 or 2 in the channel, one
to
soma, and you get propagation.
On, the propagation on the tissue, on the other compartment. But just to be clear, we don't do it yet.
We haven't tested it. It's
yeah.
feasible with the technology.
Yeah. It's an idea. It's a project we want to open, but it's not. We don't do.
Okay.
Yes.
So another question, thanks guys. Another question, because we have questions and questions and questions. We have a lot of questions. Thanks very much for the interactivity, by the way, from the audience. The question still around the electrophysiology. Did you assess as a control whether sensory neuron exhibited a decrease in spontaneous firing rate after being co-cultured with keratinocytes? So. So the question is, did you assess as a control whether sensory neurons exhibited a decrease in spontaneous firing rate after being co-cultured?
Well, we haven't done very long-term experiments in co-culture. What I can tell is when we do short-time experiments at 24, 48 hours after the beginning of the co-culture, we do not have decrease in spontaneous activity. It is possible that it happens later on. We do not have the data on this yet.
Okay, so there are some questions regarding the cosmetic and skin application. In this experiment, the one probably you have described, Alexandre, are you adding compound to the keratinocyte, to the neuron directly? And how are you adding the compounds during recording? So we're talking about compounds.
So it depends of the graph I showed you. It was on neuron alone. We did experiments with neuron plus keratinocytes. So we put, if in this context, we put the drug in the channel 3, so in the channel with keratinocytes and the axon ending, obviously. So not we can put the treatment only on neurons if you need also, but in our context we put on the keratinocytes. And how are you adding the compound during recording? How we do it? We just use a pipette, and we open the machine, and
we leave the machine open, and we add during the recording
with pipette.
With the pipette, yes.
It's the inlet and outlet reservoir of our chip to seed or to put treatment are the same as you have in P96 well. So you use it as easy. In the same manner as you use a P96 well. So we open the plate, we put with the pipette. So we have a specific protocol to not add to roughly the medium in order to decrease, to avoid the flow effect, as I showed you. So we have a specific protocol, but we provide a protocol with all our chips when you buy chips. So we have a specific protocol, but it's just the speed. Control the speed.
Yes, you have not to touch the machine, otherwise you create artifacts and everything. But it's quite straightforward in the end. You just pipe it inside.
Okay, thanks guys for the answers. There are still another question about cosmetology, dermatology application. I am interested in how the innovative skin model can be used to model inflammatory pain signals. For example, inflammatory mediators that would be derived in the skin and evidence of a pain signal via electrical signals of the sensory neuron. Can you talk about how this could be set up? I don't know if it's a big and long answer, then we may have to answer to the
It could be a long answer, but very interesting. So we could maybe be in contact directly with this person. The idea is actually we can either recreate, if the idea is to recreate an inflammatory model, first we start with a healthy model, and then we have different way. Either we put a cocktail of cytokine in the keratinocytes compartment in order to recreate the disease, or we can imagine we didn't do it, but we can imagine maybe to have already keratinocyte cells from donor with the disease. For example, if you're interested about psoriasis or eczema, we can imagine to have primary cells, a biopsy from keratinocytes from this patient and put the keratinocytes inside. It's something, it's a true way to recreate an inflammatory or injury model. And then you can also, if it's a part of the question, but you can also sample the supernatant to follow the cytokine, pro-inflammatory cytokine released, for example, by your model. And if you want to assess the effect of anti-inflammatory product.
If I can add something, so you talked about the keratinocyte side. You can also imagine something that we haven't done yet, but that you could imagine overstimulate with the neurons to generate a kind of inflammatory response on the keratinocyte side. So by using neuromediators, by using different activators, you could
electrical stimulation. Why not?
Electrical.
To mimic, actually, in this sense, we can mimic exactly the rash of psoriasis. The rash comes from stress. We know eczema. It's a skin disease, but it's mediated by the psychism. So we know that some rash are triggered by stress. So the stress comes from the brain. So by mimicking, by using electrical stimulation on the cell body of the neuron, we can imagine to recreate this rash. Could be very nice, scientifically and speaking, be very, very interesting.
Thanks, both of you guys. I think I cannot take maybe all the questions. Let me take another questions because time is running. We still have a few minutes, but not so much. So there was a simple question. What is the medium volume fitting in one channel? So we can say it's around 120 microliter, more or less, for the channel 1 and channel 3. And the channel 2, if it's tree culture, it's roughly the same, or if it's the channel 2, which is in DuaLink, where the channel 2 is very small, then we have much less volume.
Just in addition to this, because the question behind this one is it enough to do ELISA? I think. Okay. So the answer is yes. If it's a question behind this, you can do ELISA. It depends on the kit, but some kit, for example, substance P, you need only 50 microliters. You can also use multiplex, and in multiplex you can with one sampling, so 120 microliter, you can do till five mediator. You can assess, you can dose 5 mediators with multiplex, and we are developing another chip which will have more volume. But it will be another surprise.
It's really wonderful. So thank you very, very much for the interactivity. We have not been able to take all the questions, but however, we do have, of course, the person that have asked the question, so we will respond to that by tomorrow morning, and we will get also a replay of this webinar. We will send it to you, and finally, I would like to talk to you about 2 things. First, the next sessions of webinar. The next one will be the 25th of April. We will talk about CNS applications, especially in the IBD, but also we will talk about cerebral organoid culture. So you will also hear about 3D culture and 3D architectures that Netri has. We will do then a CIPN, pain on MEA, let's put it that way.
Development of
at the end of May. And it will move forward every month. So you have been nice enough to stay. The good news is that the ones that are still listening here, and I hope you are so many, then we have specific discounts on architectures that we will forward to you, that you will be able to look at if you have some interest in buying devices then you will be able to benefit from it. So it's going to be a good bargain. That's just purely commercial, but I think it's also a way to reward the people that have been very interactive in this session. So thank you very much, and we look forward to continue the discussion after the webinar, and of course to see you at the next webinar also. Thank you very much.
Bye.